Well, electricity for stress is really common. And then on the final exam, the unit portion, would it be heavy on chapters 19 through 20? The ones who are doing this, would be heavy on Bible more? I mean, that's literally all the teaching.
For the,
Unit 4 stuff.
yeah, unit 4 is 19 through, it's gonna be 19 through 24, like pretty equally, yeah. So 19 through 24, that's why I'm gonna post the review for 23 and 24, so that you guys don't miss out on that stuff, right? But you still, you know, technically have to know it just as much as you would 19 through 22. But yeah, so unit 4 is
The
that.
last exam, there's like one prominent chapter. It's any of this stuff.
Yeah, so no, not really, I don't have anything to say about that this time, yeah. It's pretty evenly spread, yeah. It kind of makes sense, like as you're going through it, you kind of get that sense of it. Some diseases stand out more than others, right? So things like tuberculosis, HIV, meningitis, like it focused a little bit more on that in each of those chapters that they responded to or corresponded to, but yeah. Okay, let me bring this up. Let me share my screen first. Share. Make sure I have audio on. I do. Yeah. Okay. It's going to be a long one. We kind of know that now by now, though. So chapters 19, 20, 21, and 22. Chapter 19 was the skin and eye diseases. Chapter 20 was the nervous system diseases. 21 was diseases of the cardiovascular system, and 22 is diseases of the respiratory tract. So we're kind of going to go over the gist of the major diseases associated with each of these chapters. First, to remind you guys, in chapter 19, talking about the skin and eye, the skin and the eye both have a very similar microbiota. So they do have an intrinsic microbiome associated with them. And their microbes that populate those biomes are very similar. For the skin, this also is dominated by Staphylococcus epidermidis. So that's something to be aware of there. Some defenses of our skin. So this is what we would say with our immune system. We talk about the first line of defense, the second line of defense, and the third line of defense. The first line of defense is stuff like this. A second line is your innate response, and the third line of defense is your acquired or specific immunity, your T cells and B cell, and the memory that comes with them. So the built-in defenses, the first line of defenses for our skin and our eye. We have high salt concentration in these areas, a low pH, lysozyme, which is present in sweat as well as in tears. And lysozyme is an enzyme that breaks down peptidoglycan very nondescriptly, and keratin, which is a waterproofing protein. And we learned about these a little bit in our innate immunity, and that these are giving some protection to these areas. The resident biota will include, why do I always do this? The resident biota will include, like I said, Staphylococcus epidermidis, as well as on the skin, Propionibacterium acnes, which is associated with acne, like it sounds. All right, so let's get into our diseases for this chapter. First, we started off with, I do it every time, and I can't zoom in without it making me very frustrated. Anyways. I'm trying. Okay, so Streptococcus pyogenes and Staphylococcus aureus were our two main biggies at the beginning. Both Staphylococcus aureus and Streptococcus pyogenes can be associated with impetigo. Staphylococcus aureus was also associated with cellulitis, which was talked about in the chapter, as well as, like I mentioned previously in the lab, Staphylococcus scalded skin syndrome, which is essentially a response to toxins associated with Staph aureus in the skin, causing the skin to sort of separate. These are due to virulence factors, which are kind of shared between both Staph and Strep. But one of the virulence factors of staph that allows it to cause these skin diseases is the exfoliative toxins. And they do what they say, that they sound like they do, cause the skin to exfoliate from itself. So it causes the skin cells to separate from each other. These guys also produce enzymes like hyaluronidase, which is an enzyme that breaks down hyaluronic acid, which is important in our extracellular matrix, or collagenase, which is an enzyme that breaks down collagen, also important in our extracellular matrix. And those help these organisms get into deeper tissues. Streptococcus pyogenes, yes, we're going to learn about how it causes strep throat in our respiratory chapter, which we'll also be going over today. But for skin diseases that it's associated with, impetigo, oftentimes impetigo starts with strep pyogenes, and then staphylococcus sort of like takes over. And staph can actually push out strep from growing because staph is more effective at growing in those environments when we're talking about impetigo. Started by strep and then grown over by staph. Strep is also associated with necrotizing fasciitis. Necrotizing fasciitis can be caused by a lot of different bacteria, but one of the more prominent ones will be strep. Necrotizing fasciitis, again, we have necrosis of tissue. We have death of the tissue associated with that and decay of the tissue. And it's pretty fast progressing. Oftentimes, major way of controlling the spread of necrotizing fasciitis will be debridement, which is removal of the damaged and decaying tissues, as well as potentially even limb loss and removal of healthy tissue to try to beat the infection before it spreads. Scarlet fever is where we have the rash associated with strep throat infection. If you or your kid ever had strep throat and got a rash on their body, that is scarlet fever. That expression of that rash associated with the infection is caused by toxins that we call erythrogenic toxin, erythro being your red blood cells. And this causes presentation of that redness and that rash due to this toxin. And strep has this toxin thanks to lysogenic conversion. It has been lysogenized by a phage, which is a virus that infects bacteria. And that virus, when it infected strep, gave it the ability to produce this erythrogenic toxin. This is actually true for a lot of the toxins that our bacteria produce. And the rheumatic fever associated with strep infection, this isn't really a skin-associated thing. This is associated more with the heart and can cause inflammation of the heart and vasculature because strep pyogenes, its capsule, looks a lot like a lot of molecules in our own body. And so when our own body tries to attack strep, it ends up instead attacking our own body. And that can lead to rheumatic fever. Usually these things can clear up and get better once the infection has cleared. And it might take some time, but they usually go away on their own, associated with clearing of the infection. All right, moving on to gas gangrene. Gas gangrene is an anaerobic infection. It's caused by the bacterium Clostridium perfringens, and we know because it's a Clostridium organism that it is an endospore former. The endospores will get deep into the tissue where there's no oxygen. This is usually due to a puncture wound, deep puncture wounds, or something like that. However, they can also grow in the gut and make toxins that are associated with GI food poisoning. This is also why we'll talk more on the GI stuff. That'll be chapter 23. That'll have a video about it. But Clostridium perfringens found in a lot of places in the environment, including naturally harbored as part of our biome in our vagina or even in our gut. But when it overgrows in areas where it doesn't, or it gets to areas where it doesn't belong, is whenever we see presentation of disease. So in the case of gas gangrene, we get this a deep tissue infection where there's no oxygen. And this is an anaerobic organism that produces gases as a byproduct of its anaerobic respiration. Those gases build up in the tissues and cause pressure under the skin. The best way to relieve that pressure will be through fasciotomy. So we'll actually cut open the skin to relieve the pressure from the swelling of the gases and the infection and the inflammation and everything like that. Treatment for wounds like this or prevention of gas gangrene will be deep tissue flushing with hydrogen peroxide. These guys don't make catalase. They don't have any sort of way to protect from that sort of damage. So treatment with that sort of oxidizer reagent will be effective for treating that. But once it becomes more deeply seated in the tissues or has spread a little bit more systemically, then treating that will have to be done with more antibiotics and more focused targeting like that. All right, chickenpox. Chickenpox is caused by the virus varicella zoster. It is a herpes virus. It's also known as human herpes virus. There's several different kinds of human herpes virus, HHVs. I believe this one is human herpes virus 3. It's either 3 or 7. I'm not going to test you on that necessarily, but it's a human herpes virus. So it is a herpes virus, and all herpes viruses are DNA viruses that will become latent in their hosts. And so they will hide out usually in your nerves, like all herpes viruses do, and basically maintain a provirus, which is their DNA version virus, inside the DNA cells of the nervous system. And then when you're under stress or when your immune system wanes, they can come back out again. We don't see chickenpox doing that a lot as frequently as what we see with herpes, HSV infections, but it can happen in the form of shingles later, usually more commonly later in life, in your 50s approximately, though people can have recurrence of this earlier in life than that. Usually whenever we have reactivation of chickenpox in the form of shingles, it comes out along whatever nerve it was hiding out in, often one of the dorsal nerves. So we usually see shingles presenting on like the trunk that way. So it is a herpes virus, and once you have chickenpox, once you've gotten it, you have it for life because it hides out in your body, just like every herpes virus does. Next is smallpox. Smallpox is caused by a different virus. This is a pox virus, a true pox virus. And this is the virus that causes smallpox is called variola virus. There's 2 different kinds of variola, variola major and variola minor. The one that we more commonly think of with smallpox infection is going to be variola major. We do have a 30 to 50% mortality rate associated with smallpox infection. This is the number of people that die from getting smallpox. So about 30 to 50% of the population that gets smallpox will die from it. It usually leads to initial presentation with fever and malaise and is more commonly known for its disfiguring rash. The rash can cause permanent scarring regardless of whether or not you are scratching or anything like that. With chickenpox, the rash that's associated with chickenpox, you can recover from it without any scarring. You only get scarring if you get secondary infections. Usually secondary infections are going to be caused by staph and things like that. So I have plenty of chickenpox scars, let me tell you, because my mom let me scratch it with my dirty little fingers when I had the chickenpox. But anyway, smallpox will cause a disfiguring rash that will cause scarring that is permanent, regardless of scratching or anything like that. We've eradicated smallpox from the planet. Smallpox does not exist anymore on the planet. This is thanks to the very first vaccine that was ever invented. It was using cowpox, which is a very similar related virus, and that virus is called vaccinia, which is why vaccines are called vaccines. Vacca, the Latin for cow, was cowpox vaccinia. And having an infection with cowpox led to protection from smallpox. And so that was the very first vaccine, originally created by Edward Jenner. It's a rough story, man. He took his kid and inoculated his kid and then pushed his kid out into smallpox population. And his kid didn't get it. And basically all smallpox vaccines were essentially just modified and improved from that same idea. We do keep stores of smallpox in several countries across the world, including here in the United States. The CDC keeps a frozen store, as well as the Army keeps a frozen store. There are other countries across the world that keep frozen stores of smallpox. Even though this is a disease that we haven't seen at all since at least the 70s, nobody has had this disease at all. We still have plenty of countries vaccinating against this. The United States does not vaccinate against smallpox, except for in our military. The military does vaccinate against smallpox. Russia and most other countries outside of the United States still vaccinate for smallpox because of the risk for bioterrorism. And so it's really the only reason why other countries still vaccinate for that. Next is measles. We learned a little bit about the measles already throughout our talks throughout the course. But measles is caused by the measles virus, pretty straightforward. It does cause a rash. It's a maculopapular rash. It's a raised rash with some coloring to it, which is what makes it maculopapular. So you will have spots associated with this. It is a viral disease, and we do have very effective vaccines against it. And the best way to protect against it is through vaccination. 1 in 1,000 people that get the measles will have encephalitis, and many of those people die from having encephalitis. So it ends up being about 1 in 1,000 people who get measles end up dying from it. That's not a small number when you consider this is the most contagious disease that mankind has ever known. So it's highly contagious. We have even common cold, less contagious, even COVID, less contagious, even flu, less contagious than the measles. For the reason that we don't see the measles in the populations, I will reiterate it one more time, one last little dying breath in me. The reason we don't see measles in our population is because people have been generally vaccinated against it. So next is ringworm. Ringworm, despite its name, worm, it is not caused by worms. It is caused by fungal infections, and they'll often present in a ring shape. These are fungal infections. Another word for a fungal skin infection is a cutaneous, cutaneous being the skin, mycosis, mycosis being a fungal infection. So these cutaneous mycoses, which is the plural, this includes not just ringworm like we typically would see on the skin, which would be Tinea corpus, but even in the beard, Tinea barbae, or Tinea pedis, which is athlete's foot. There's all sorts of different versions of it wherever you get it on your different body, but they're all caused by a group of cutaneous fungal groups, and these are called dermatophytes. They are not caused by worms; they're caused by fungi. Next is the eyes diseases. We talk about conjunctivitis. The conjunctiva is the protective layer over our eye tissues, and that can become infected, and as a result of the infection, become inflamed. Anytime that we see itis anywhere, anytime you guys see itis, itis means inflammation happened. It doesn't necessarily mean infection. You can have inflammation without infection. But if you have itis, we have inflammation. So conjunctivitis, inflammation of the conjunctiva, the outer layers of the eye, can be caused by bacterial infection, viral infections, or from allergies. When you have allergies, you'll have a lot of clear fluid and exudate, and you'll usually have a runny nose and things associated with the allergies. Viral infections, they will be pretty similar in presentation to allergic, just with less fluid, clear fluid still, but less. The eye will also be red as a result of the inflammation. But bacterial conjunctivitis, you will have a milky or white discharge, very creamy looking discharge associated with all the bacteria growth in the area, as well as our white blood cells that are reacting to it. The eye is an immune privileged area, so we have less of an immune response. When we talk about immune privileged, these are tissues like the eye, as well as our central nervous system. Immune privileged site like the eye and the central nervous system have less immune response. And this is because the inflammation that our immune response causes can cause damage to those tissues. And if you were hunter-gatherer back in the day, you couldn't afford to lose your eyesight. And if you lost nervous system function, especially in the central nervous system, we're talking permanent damage. So we have less of an immune response in the eye, as well as a significantly less in our central nervous system. Those are immune privileged sites. And so we do have a different sort of immune response going on in eye infections. All right, so that's conjunctivitis. It also talked about keratitis. So that would be actual inflammation of like the cornea itself. So the windshield of the eye, the outer coating of our eye. So when the cornea becomes infected and inflamed, that is called keratitis. And that's usually going to be associated with transfer of herpes simplex virus 1 to the eye, or from acanthamoeba, which is an amoeba. It is a protozoa. Due to contaminated water. So if you are rinsing your contact lenses with tap water, never do that again for the rest of your life, please. Always use sterile saline for that. And you want to be sure it is sterilized, because acanthamoeba can live in tap water. It can live in your hot water tanks. It can live in your pipes. And you do not want to get keratitis, because that can actually lead to permanent damage to your eye or even blindness. And it's pretty difficult to treat. You've probably even seen Instagram videos about people that have had that happen before. So avoid keratitis that way. Keratitis can also be caused by herpes simplex virus, like I said. HSV1, herpes simplex virus 1, is the one that's associated with cold sores. It's oral herpes, more commonly with oral herpes. When you have transfer of oral herpes to the eye is whenever you get keratitis associated with that HSV, and that can cause damage as well. All right, so trachoma is our next one. Trachoma is a disease caused by the bacteria Chlamydia trachomatis. Chlamydia trachomatis is, like it sounds in the name, also the chlamydia that is associated with the sexually transmitted chlamydia. It's the same bacteria, but we're talking about different transfer, different infection methods. So trachoma can actually transmit from like basically fecal to eye contact, contaminated surfaces to eye, and it's usually associated with poor sanitary conditions and can even be associated with fly landing on poop and then, landing in your eyes and stuff like that. Trachoma is the leading infectious cause of blindness in the world, which should say something to us in our lack of understanding of underdeveloped countries because we don't see trachoma here in the United States, but we see this a lot in underdeveloped countries. So worldwide, it's a huge issue. Next is river blindness. River blindness is caused by a helminth. This is the helminth Onchocerca volvulus. This is a parasitic worm that gets into the eye, usually through transmission by flies, and it can even migrate through the tissues and then into the eye itself. And so the worm actually lives inside of the eye, and it causes what we call river blindness. The reason that the worm is pathogenic is because it's also infected. The worm has a bacterial infection. It is infected by the bacteria Wolbachia, and many, many helminths are infected by Wolbachia bacteria. We call this an endosymbiont. This is a bacteria that has created a symbiotic lifestyle with our helminths and then makes our helminths more pathogenic. This is true for heartworms with dogs as well. My dogs had heartworms, and we had to treat them with ivermectin. And so many of these helminth infections that are treatable with ivermectin, we often will also treat with doxycycline, because doxycycline, being an antibiotic for bacterial infections, will kill the Wolbachia endosymbiont and make the worms weaker. Next is chapter 20. Chapter 20 is our nervous system diseases. The nervous system, peripheral or central nervous system, does not have a biota. No biota. No biota for the central nervous system. It should be sterile. And like we said, it is an immune privileged site. Your nervous system is immune privileged, especially your CNS, because your central nervous system can't regenerate the nerves. So this includes the covering of the nervous system called the meninges, the dura mater, the pia mater, and the arachnoid mater. I think that's right. So anyways, those three layers of the meninges, which is the outer covering of our central nervous system. And that can become infected. And then your actual brain and your nervous tissue can also become infected. And they cause different kind of presentations and different kinds of disease. So we'll start with meningitis. Remember, itis is inflammation. So this is inflammation of the meninges, and we're talking about infectious causes of meningitis. We have three main categories of infectious meningitis: bacterial, fungal, or viral. Can you have meningitis caused by other things that, like, for example, potentially a protozoal infection? Yes, but these are our main ones that we're talking about here. Okay. So first one, the bacterial meningitis. The most common cause of bacterial meningitis is going to be Streptococcus pneumoniae. Streptococcus pneumoniae, also known as the pneumococcus. We do have a vaccine for pneumococcal meningitis. It also protects from pneumococcal pneumonia. Because as it turns out, Streptococcus pneumoniae, it doesn't just appear here, does it? It appears in a lot of places in these chapters. Streptococcus pneumoniae, as the name suggests, is also the #1 cause of pneumonia. So it's the #1 cause of meningitis, bacterial meningitis as well. And there is a vaccine for it called the PCV vaccine. It is a gram-positive, and as its name suggests, it is a streptococcus. So we're talking chains of cocci. It can be in pairs or chains, but it is gram-positive. So this is really important to remember. The worst kind of bacterial meningitis that you can get is caused by Neisseria meningitidis. And so this one is known as the meningococcus. This is transmitted through close contact. It's not really going to be an issue if you're not living in close living quarters with a lot of people, but we see this a lot in dorms and barracks and things like that. It causes a petechial rash. So we do have a rash associated with this red spots on the body. It is petechial. This means that the rash is actually caused by tiny burst capillaries under the skin. The capillaries under the skin are bursting because of the toxins associated with Neisseria meningitidis, including, maybe most importantly, endotoxin, because Neisseria meningitidis is a gram-negative diplococcus. And this is important to remember as well. So already, if your patient comes in with meningitis, we've got, just do a gram stain on their CSF fluid, and we're starting to see enough information already. So let's go through our other guys. Haemophilus influenzae. This guy is also known as Hib, Haemophilus for H, influenzae for I, and it's serotype B is the more common cause. So this is also associated with bacterial meningitis. And then we have Listeria monocytogenes. So I will say this, Haemophilus influenzae is also gram-negative, but it is not a Diplococcus. And Listeria monocytogenes is a gram-positive bacillus. It's technically a coccobacillus, but it's definitely leaning towards bacillus, okay? And is also motile. Whereas Haemophilus influenzae is non-motile. So we've got a lot of defining characteristics that we can already sort out basically what these guys are just with a gram stain. Just with a gram stain, we can determine the difference between all of these 4 bacterial guys. Also, if you do a gram stain, if your patient has meningitis and they have a fungal infection instead, what would you see differently on a gram stain? So we don't typically do gram stains on fungal specimens, right? But fungal species will have cells in the gram stain. They'll still stain with the gram stain. But what we will see is a nucleus. Whereas bacteria, no nucleus. And much smaller. All right. So our 2 versions of fungal meningitis that we can sometimes see are either caused by Cryptococcus neoformans, which will be the more common one. We usually don't see this very often in people who are otherwise healthy. This is usually associated with immune compromise. And cryptococcal meningitis is usually transmitted via bird droppings. We see this a lot in homeless populations. People that already have tuberculosis or HIV are going to be more susceptible to this stuff. Another one that can cause meningitis is coccidioides immitis. It's more commonly associated with the disease valley fever. There are other symptoms associated with valley fever, but it can progress into meningitis. Rarely, but it can. And then viral meningitis. This by far is the most common type of meningitis. Most cases of meningitis will be viral, and 90% of viral meningitis cases will clear up on their own. They won't need treatment. So we don't talk about it very much. But if you have a patient that has a stiff neck, photophobia, which is light sensitivity, maybe they've had headaches, and there's something called Budzinski's sign, which has to deal with when you bend their knees up and then bending their neck and stuff like that. So if they have, and fever, usually presented with this. So if they have these signs of meningitis, first thing you're going to do is, number 1, start them on antibiotics, regardless. You're going to start them on antibiotics for Neisseria meningitis, just in case it's that, because this is horrible. This is a horrible disease that can become systemic very quickly and kill you very fast. So you aren't going to care if it's bacterial, fungal, or viral at first. You're going to say this is presenting as meningitis. Start them on broad spectrum antibiotics, definitely targeting for Neisseria meningitis. Then you're going to get your gram stain of your cerebrospinal fluid, the fluid that is surrounding our nervous system. And then have the lab do the gram stain on that. The gram stain will tell you. If you look in the CSF and you do a gram stain of the CSF, if you don't see any cells, there's no bacteria and there's no fungi, but they're presenting with meningitis, it's viral. You don't need to see anything on that. So it's what we call aseptic meningitis. You won't see anything in the CSF, but they're presenting with meningitis. They probably have viral meningitis. If you look in your gram stain and you see big cells with nuclei, it's fungal. And if you look in your gram stain and you see tiny cells that are paired up and they are gram negative, then it's Neisseria meningitis. If you see tiny cells that are in chains or pairs, but they're gram positive, Streptococcus pneumoniae, and so on and so forth. So you can just do a gram stain of the CSF, and that will help you target down your treatment a lot better. You can see why we cover this a lot. And yeah, I bet that you're going to talk about this in the nursing program if you guys haven't already. So next, we have meningoencephalitis. Meningoencephalitis, when we get into encephalitis, this is the in-between one. Meningo is going to be your meninges. Encephalitis is your actual brain tissue. So meningoencephalitis, you have inflammation of both the meninges and the brain tissue at the same time. And it can be caused by a number of things, but the two that we're focusing on are both amoebas. They are amoebas. And I'll remind you guys, an amoeba is a protozoa. All protozoa are eukaryotic single-cell organisms. And so these amoebas, if we know they're an amoeba, we know that they're a protozoa that moves by those pseudopods, right? They use the extensions of the cell to walk like little feet to pull themselves along, like we think of with amoebas. So these are both amoebas, and there's two different ones, Naegleria fowleri and acanthamoeba, which I already mentioned acanthamoeba in our keratitis talk just a minute ago. Both of these guys can live in warm water. Naegleria fowleri as a protozoa can live in both warm water, freshwater bodies of water, especially when it's warm. So we're talking ponds and lakes. That's usually standing freshwater, but it can also live in your hot water tank. Same with acanthamoeba. You're more likely to be exposed to acanthamoeba from your own fresh tap water than you are Naegleria fowleri. But more commonly, if people have Naegleria fowleri infections, about half of those infections are going to be associated with swimming in fresh water, and the other half is going to be from tap water exposure. This is why I say never use a neti pot with tap water. If you're going to use a neti pot with tap water, boil your tap water for 30 minutes at least, then cool it down to the temperature that you like and mix in your buffering agents like salt and stuff like that before you neti pot with it, because otherwise you could be introducing Naegleria fowleri or acanthamoeba into your sinuses. Once it gets into your sinuses, it's a pretty easy jump for them to make it to the brain. These are extremely rare infections, but they are 97% fatal, 97 to 98% fatal. The people that do survive, the other 2 or 3% that survive these infections, will have deficits. You do not survive and go on and walk a normal life from these infections. They're pretty quick acting. There is no treatment for this. Once you start having these signs and symptoms, it's pretty much a death sentence. So this is the brain-eating amoeba, right? That's what this is. Naegleria fowleri presents as primary amoebic meningoencephalitis, or PAM, whereas acanthamoeba presents as granulomatous amoebic meningoencephalitis. And so granulomas are whenever our white blood cells try to wall off infections. And so we see that as a characteristic of acanthamoeba infections in these circumstances. All right, moving on to encephalitis. Itis being inflammation, encephalo being inflammation of the brain. So we don't necessarily have meningitis associated with this. If you get one of these infections, can you have meningitis associated with it? Yes, but we're talking about the presentation specifically of the encephalitis. The number one causes of encephalitis, well, the number one overall cause of encephalitis is going to be herpes virus, which we don't even talk about in this chapter, but it is the number one cause of encephalitis. But next to that easily is going to be our arbovirus group. And so arboviruses are very commonly associated with encephalitis. An arbovirus is a virus that is transmitted by a biting insect, any biting insect, but the ones we talk about the most in this chapter are ones transmitted by mosquitoes. And so this includes West Nile virus. This includes Zika. This includes any of those arboviruses that can be transmitted by biting mosquitoes. And even I told you guys, I'm pretty sure that my husband had West Nile recently. He was having like neck pain and his head was hurting and then his back started hurting. And I'm just like, you baby, get over it. Like you're fine. You have a headache and your back hurts. You've been working. Of course your back hurts. And then later he had this petechial rash all over his legs. And I was like, oh, okay. Well, maybe you're not a baby after all. And we had recently, a week or two prior, maybe a week and a half prior to that, our local park, which is Edmund Lyon, because I live in Bethany, they had delayed fireworks and we could see them from our front lawn. We were sitting in our front lawn watching the fireworks and he was getting eaten alive by these mosquitoes. And so it tracks. And so he probably had West Nile virus. Many of these arboviral infections, many of them, you will not have signs or symptoms of, or they'll be subclinical to the level that you would not think about going to the doctor. Having a little headache, having your back hurt a little bit, do you go to the doctor for that? Most of the time not. And so we call that subclinical. Once we get to the point where we're having a fever or we're actually getting, inconvenienced by our pain or it's significant causing us from not getting enough sleep, that's usually when we'll go to the doctor. So many cases of our arboviral infections aren't going tracked. So we might think not many people in Oklahoma or the US are getting West Nile, but how many of them are subclinical or how many of them are asymptomatic? They're not going to the doctor, it's not getting documented. So West Nile and Zika and things like that are actually probably considerably more common than we realize. It's not getting tracked. So includes stuff like that. And there's some more deadly ones like equine encephalitis virus and stuff like that. But they're all transmitted by mosquito and can lead to encephalitis, which is inflammation of the brain tissue. Signs and symptoms associated with encephalitis specifically, you might have headache, you might have fever, confusion, even personality changes. So actual neurological effects that could affect the brain can accompany encephalitis. So let's get on to our more bigger categories of our encephalitis. So for rabies and poliovirus, both of these are acute. With the prions, this I would say is subacute. All right. So acute because we're having significant presentation of symptoms. It's a little faster acting and very severe effects from these infections. Whereas the subacute with the prion one, which we'll get to in a minute, we have slow progression of the disease and more mild presentation until it kills you, and then it doesn't seem so mild anymore. So starting off with rabies. Rabies is a viral disease that can cause encephalitis. It is caused by the rabies virus, which is a bullet-shaped virus. It is transmitted in the saliva, typically, and went from the saliva through a bite can move into the nervous system. It takes it a while to migrate into the nervous system from the tissues, but once it gets into the central nervous system and we start having signs and symptoms associated with rabies, there's nothing we can do for anybody, animal or human. Therefore, we say once rabies has established itself as an infection and has actually caused disease, once we're in the prodrome and leading into the acute phase of the disease, but once you're in the prodrome, which is our initial signs and symptoms, rabies is incurable and is 100% fatal. So what happens is you get bitten by an animal, the virus can multiply in any tissue in your body, and it will move gradually into the nervous system. So in between the bite and the moving to the nervous system, we have a window that we can treat. And so for humans, that means giving immunoglobulin that has antibodies from other people that have survived rabies that is protective against the rabies from that. And so we would call that an artificial passive immunity. But we'll also give what we call a prophylactic post-exposure vaccine. And so we'll vaccinate you against rabies as soon as we can as well. And that should elicit your own active response. So, we call it artificial active as well. So we have artificial active and artificial passive protection from the immune response against rabies. No antivirals really are going to help with this in time. It does take about a month for signs and symptoms to present. So 3 weeks to a month for signs and symptoms to show in rabies. But once you have that initial signs and symptoms, there's nothing we can do. It's way more common in animals, of course, and especially in wild animals. Vaccine efforts are underway. We do spread vaccine treats throughout the wild to try to even vaccinate wild animals, and it's been pretty successful overall. Poliovirus. Poliovirus is another viral infection. Polio exists only in humans. No other animal on the planet can have poliovirus. So this is a disease that exists and sustains itself only within humans. The poliovirus is a very, very tiny virus that is what we call a naked virus. It does not have an envelope. It is just a capsid and nucleic acid. But despite that, is one of the most affected viral infections that you can get. It's transmitted through fecal oral contamination. And so people were getting drinking water that was contaminated with this virus. It's an enterovirus, meaning that it gets into your enteric system, which is your gut. You swallow it, it gets into the gut, and then eventually moves into the central nervous system where we have the typical display of polio. Nervous system degradation, often loss of muscle function associated with it, which can even be permanent and lead to permanent paralysis. Poliovirus is well known for its sequelae, the sequelae being the lasting effects even after you have cleared the virus. So poliovirus can lead to paralysis. And many cases, people who were infected with poliovirus even wound up in iron lungs, which was basically just a big tube that you'd be put in, and there was a bellows on it. And the bellows would basically cause pressure to build up in the chamber that would cause your body to breathe in and out because you could no longer control your diaphragm. And if you were taken out of the iron lung, then you would suffocate because you couldn't breathe on your own anymore. So we have pictures of rooms and rooms and rooms of people that were living in iron lungs that were breathing for these people. They were fully conscious, you know, they weren't unconscious or anything like that, but they're just paralyzed from this disease. Horrible disease. It's been mostly eradicated from the planet thanks to vaccination efforts, but it still exists in Afghanistan and Pakistan, so it's not completely eradicated. We still have it in Pakistan and Afghanistan. All right, so moving on to our subacute encephalitis. There was prions, of course, which I'll get to in a second, but there was also toxoplasmosis, which was another one. Toxoplasmosis, which was in our notes, caused by Toxoplasma gondii, is a protozoal infection. Toxoplasma gondii, it exists in feline populations. It's where the natural carriers, the natural reservoir is cats. And they can transmit the protozoa through their feces and their urine. And then mice can become infected by being in the environment. And what happens is when you get infected with Toxoplasma gondii, in people too, but it's been very well documented in mice, that they have an increase in risk-taking behaviors. They're more likely to present themselves. They're more likely to appear in front of cats and things like that, that could potentially, you know, want to eat them or whatever. And so the cats will eat the mice and then become reinfected, and we're just propagating the cycle like that. Studies have shown that people who are infected with Toxoplasma gondii might also present with risk-taking behaviors as well during their infection course. So pretty interesting stuff. When pregnant women become infected with toxoplasma, especially during the first trimester, it can lead to abortion of the fetus. It's about 30% chance. So if you are a pregnant woman and you have cats, do not handle the kitty litter. It's also advised people who are on immunosuppressants or people who are going through chemotherapy or radiation, they also have lowered immune systems, should also not be handling cat litter for the exact same reason. In those people, they're not likely to lose their fetus, because they're not pregnant necessarily. But if you have a lowered immune system, then you're going to be more susceptible to the effects of the encephalitis. And so the actual inflammation in the brain and the disease associated with having Toxoplasma gondii, which is really a subacute encephalitis, but can be dangerous in immunocompromised individuals. And 30% of cases can lead to stillbirth or even abortion of the fetus. Okay. The prions. Prions are just misfolded proteins. And so it's a normal protein that's in your body, which we call PRP. Normal protein in your body, the PRP protein. But it becomes misfolded. So it has a normal function in your nervous system and whatever. If something happens to cause it to be misfolded, it doesn't fold the way that it's supposed to in its normal functional state. And when it becomes misfolded, a lot of those hydrophobic groups that were hidden on the inside become put on the outside. And so they don't mix well with your body anymore, and they just stick to each other. And so they cause these clumps in the brain that eventually lead to creating holes in the brain tissue. And so we call this a spongiform encephalopathy. Spongiform because of the holes being formed in the brain tissue. Encephalopathy, encephalo is brain, pathy is disease. And so we usually don't see the presentation of the spongiform aspect until after the patient has died and we can do an autopsy. It's a pretty difficult one to diagnose as well. We don't always see the misfolded proteins in the cerebrospinal fluid. Not to mention, there's only like one lab in the United States that can test for this in Los Alamos. And so it's expensive to get it tested and things like that. Usually you'll be exposed. What can happen is you can either have a genetic predisposition to your proteins just misfolding naturally, or you can maybe ingest something or be exposed to something during surgery to a misfolded protein, and then it causes yours to misfold the same way. We do think that there are genetic aspects to this. You are more likely, if you have certain genetic markers, to have your protein misfold, but we don't understand this entirely, why it happens or how it happens. It is pretty rare, but there was an outbreak of this in the 90s in the UK, where they were feeding beef beef. Basically, they were grinding down dead cattle and feeding it to the cattle. And so the cattle were propagating this misfolded protein in their population. And then the cattle were coming down with mad cow disease. As it turns out, people can actually get that same disease. So it's a similar protein to what's in our brains. People would eat the beef and then later on present with something similar to mad cow disease. But in people, we call it Creutzfeldt-Jakob disease, CJD. If you got it from eating cattle like that, we call it variant CJD, which is VCJD. These misfolded proteins cannot be denatured from cooking. They cannot be denatured from autoclaving. They have to be fully incinerated. We can also treat with sodium hydroxide and then treat with at autoclave temperatures higher than what we normally would. So instead of 121, we will push it to 135, which we don't do normally. This is not uncommon to see this spread through dental procedures, organ transplants, and things like that. It's been documented being spread those ways as well. Just simply because we don't normally cover for this. But like I said, extremely rare. It takes about a decade to 2 decades for the signs and symptoms to start appearing. So you can eat beef and then 10 to 20 years later start having neurological symptoms. And it comes on very gradually, but usually from the first onset to death, you have 1 to 2 years. And it is fatal, 100% fatal. There's no treatment for it. And we have tetanus. Tetanus is a spastic paralysis. So it causes the muscles so they cannot relax the muscles. It is caused by the bacterium Clostridium tetani, more specifically by a toxin produced by Clostridium tetani. This Clostridium tetani organism creates endospores. The endospores are introduced into the tissues and then turn into the vegetative bacteria that makes the toxin. Once the toxin is made, then this prevents muscles from relaxing. This will lead to that spastic paralysis. This doesn't just affect the jaw, but can affect any muscle in the body. And usually people will die because their diaphragm becomes spastic and they can no longer breathe. Related to that is botulism, another neurological effect. Obviously the opposite almost, but here we have flaccid paralysis where the muscles can't contract. This is because the toxin, the botulinum toxin made by the bacteria Clostridium botulinum, that toxin blocks acetylcholine release, which is one of the signaling molecules that signals for muscle contraction. So it blocks that, so we have true flaccid paralysis. Luckily, Clostridium bacteria in general, they don't do well in air. And if you're canning foods, they don't do well in acid. So if you can your foods in acid, then they should be protected. So pickles, not going to be a problem, typically. But things like green beans and other things like that, if we aren't truly sterilizing our cans and stuff like that, or if we're not bringing up our vegetables or whatever to an acidic pH, then Clostridium botulinum can live in those cans, and the endospores can survive pretty much indefinitely. They're not going to be boiled away. So we have to go through true sterilization to protect from botulinum. All of these Clostridium species, whether it's Clostridium tetani, Clostridium botulinum, or Clostridium perfringens that we saw in gas gangrene in the previous chapter, they all live just in the environment, and the endospores just kind of can hang out anywhere. So this is a constant risk.
Is it endospore and anaerobic?
Yes, all Clostridium species make endospores, and they are anaerobic, true anaerobes. So they aren't even facultative anaerobes. They don't ferment. They don't sometimes use oxygen. They actually can't survive in oxygen. The endospores are hardy enough to exist in the oxygen, though. They're not multiplying, of course, but once they get deep in your body, and they can become vegetative again and start multiplying again. But yes, all Clostridium species are anaerobic and can make endospores. Which is C. diff, right? So C. diff is a Clostridium species, and so it's like that as well, which we'll talk about in chapter 23. I'll have the video posted for that one. All right, chapter 21, this is our cardiovascular and lymphatic infections. When we talk about blood-based infections, if you have bacteria, so your blood, I'll say this, your biome for your blood, you should have nothing, it's sterile, just like your central nervous system. Your blood and your lymph should be no biome at all. Nothing should be living in it. So if you find bacteria in your blood or your lymph, we call that bacteremia. But if the bacteria are flourishing and actually growing in the blood or the lymph, we call that septicemia. So that's the difference between bacteremia and septicemia. If it's viruses, we call it viremia. If it's fungi, we call it fungemia. So some of the diseases we talked about, COVID-19. This is caused by a virus. The virus is called SARS-CoV-2, and the disease is COVID-19. we don't consider this a respiratory disease. This is not our respiratory chapter. This is our cardio and lymph. Point that out. Does it present with respiratory symptoms? Yes. Is it transmitted by aerosol droplets and air? Yes. But I want to be very clear about this. COVID is a systemic disease. So the SARS-CoV-2 virus can live in almost any tissue in your entire body. The receptor that it uses to get into your cells is present on most tissues of the body. This is why we find the SARS-CoV-2 virus in feces, in urine, in semen, why it can affect your GI, why it can affect your nervous system. The whole reason that people lose their sense of smell and taste, that's a neurological aspect. That's not a respiratory disease, right? So COVID-19 is something that becomes very systemic throughout the whole body. It can affect almost every single cell in the body. We have different variants of the SARS-CoV-2 virus leading to different variants of the COVID-19 disease. Alpha, Delta, and Omicron, we remember back to these, and we had even more since then. And each one is worse than the next. We have a lot of people saying like, well, then why are so many people surviving so much better? Why do you think that is? And we've talked about this previously. Yet another ploy for me to say vaccines. The vaccines that were developed, like the mRNA vaccine, have trained our body not to overreact to these viruses the way that they would naturally. So our body normally would see these viruses and have basically a systemic, what we would call it, like it's a super antigen essentially. Cytokine storm is what I'm trying to say. So all the signals for inflammation, there's just so many all at once that would lead to, like a whole body systemic over response. Yes, question.
Isn't it like back in 2020, people mainly died due to respiratory failure?
Yes. So it does affect the respiratory system, but not just the respiratory system, right? So we don't consider this a respiratory disease like tuberculosis. So tuberculosis is primarily, for most people, just in the respiratory system. You can get tuberculosis outside of that, but that's way less common. For COVID-19, the signs and symptoms that kill people might be related to the respiratory symptoms and stuff like that, but it is definitely infecting the other tissues and affecting the other tissues of your body as well. And so we consider it more systemic. It's cardiovascular and lymphatic.
You like have like shortness of breathing for a really long time.
Mm-hmm. Yes, so you'll feel, so we would say the effects of COVID can be what we call long COVID, right? And so long COVID is like presentation of basically sequelae. Like you may have even cleared the virus from your body, right? But you're having a long-term effects because your body was so damaged from the inflammation, or your body has continued to keep up the signaling even though the virus is gone. And so that's usually a result of that virus causing the signaling for those cytokines, which are what mediate our inflammatory response and our immune response. And so the biggest problem with COVID really is that it causes all that signaling to happen in the cytokines. And where does that happen? In your bloodstream. And so this is a virus that is in your blood. It does cause very severe respiratory symptoms associated with it. And oftentimes the cytokine storm effect in our lungs is where we see the most negative effect because that lung tissue is so susceptible to being damaged. And then having, like, we rely so heavily on our oxygen intake. That's where we see the most negative effect, but it is still causing and wreaking havoc throughout your entire body. So, which is why we consider it a systemic bloodstream related disease, even though it's transmitted by air. And even though people often die from the respiratory symptoms, it is technically systemic. So that's great thing. Wide ranges of symptoms. We all know this. Respiratory is very common. But we also have inflammatory symptoms associated with this, neurological symptoms associated with it. Many people are asymptomatic. 1/3 of the population that gets infected with SARS-CoV-2, the virus that causes COVID-19, 1/3 of the people infected with it will not have any signs or symptoms. And so people were walking around thinking they didn't have COVID because they didn't have any signs or symptoms, but they were just propagating it. And this is still true even today. So, this is true with a lot of viruses, but particularly with COVID, with how concerned we were about it spreading, that was an issue. All right, moving on. Plague. I'm saying plague very generally. When we talk about the Black Plague or the bubonic plague or the pneumonic plague, these are all just different versions of the plague, and where they are in the body determines what kind of plague you have. But they're all caused by the bacteria Yersinia pestis. Yersinia pestis is a gram-negative bacillus. So it's rod shaped and it stains gram-negative, meaning it's going to be red with the gram stain, right? It is transmitted by flea bite primarily, and these fleas are often harbored within the bacteria can be maintained in rodent populations. So the fleas on the rodents then bite the people and transmit the bacteria to the people. However, I want to be very clear about this. While getting the bubonic plague, which is infection in the lymph nodes, or the septicemic plague, which is whenever it gets into the bloodstream and becomes systemic. So septicemia, like we said earlier, it's actual true propagation in the blood itself. Bubonic, the infection is basically just located in lymph nodes. All right. Those are transmitted by fleas. But the pneumonic plague is when it migrates into the respiratory system. And when it gets into the respiratory system, it can be transmitted from person to person. Commonly used as a biological warfare, even in past, I mean, this is a super old disease, and people would throw like dead bodies from the plague over walls at other populations to try to get them infected, and whatever. It's not associated with bad smells. Like plague doctors wore those long masks that would be stuffed with flowers and herbs because they thought the bad smell was what was causing the disease, but turns out that doesn't protect them at all. All they had to do was be bitten by a flea that had this bacteria or get it from somebody who had the pneumonic plague. So those swollen lymph nodes would actually be so large under the skin, you could see them through the skin, and they would even, just, they would be huge. They'd be massive. The septicemic plague, once you had the bacteria flourishing in the bloodstream, we would have decreased blood flow to the extremities and also basically a systemic inflammatory response. Because this is a gram-negative bacteria, gram-negatives have endotoxin, LPS, all of them do. And so that endotoxin LPS was causing a shock response within the body as well. And this led to death of the extremities, the tissues in the extremities. And so we would get black fingers, black toes, and that would migrate up as necrosis set in. And so that's why it was called the Black Plague. Septicemic plague was the Black Plague. And will lead to death and necrosis of the tissues, literally turning them black. All right, moving on, infectious mononucleosis. Mononucleosis is talking about mononuclear cells. So this would be like your macrophages, your monocytes, and all of your B and T cells as well. So these are our monocytes. These are our mononucleus cells. These are our agranulocytes. And so we call it mononucleosis, but when it's from an infectious origin, that's what we consider to be mono. The kissing disease, it's caused by the virus Epstein-Barr virus, which is another human herpes virus. It can also lead to Burkitt's lymphoma, where we have lymphocytes basically becoming cancerous as a result of this virus. Because it's a herpes virus, the DNA gets integrated into your actual blood cells. And so what happens is the cells in your lymph nodes, once you've had mono, the cells in your lymph nodes harbor that DNA in those cells forever, and it can resurface. People can actually have recurrent mononucleosis as a result of that Epstein-Barr virus becoming a provirus in their own cells because it's a herpes virus, and all herpes viruses do these things. You'll see more common recurrence of mono, as well as occurrence of Burkitt's lymphoma in people who are immune compromised. You're more likely to see Burkitt's lymphoma in people who are severely immunocompromised. So this is AIDS usually when we'll see that. All right, malaria. Malaria is a protozoa. It falls into the group of protozoa called the apicomplexans. These guys are non-motile, and they are all parasites. The guys that cause malaria all are Plasmodium species. So Plasmodium something. The more common one and the more severe one is called Plasmodium falciparum, but there are other ones. So these are protozoa transmitted by mosquitoes, and it actually has a life cycle step in the mosquito gut. It actually lives and infects the mosquito itself. So you don't just need it transferred from person to person by the mosquito carrying the blood. The mosquito actually becomes infected with the malaria protozoa. We can prevent this with prophylactic treatment with drugs, chemotherapeutic prophylactic treatment. So basically using drugs, which we learned about in chapter 12, The antimalarial drugs are the ones based on quinine, so chloroquine, quinine, and things like that. We can actually treat people with those drugs to, yes, treat the malaria itself, but also give you those drugs before you go to a malaria endemic area to prevent you from ever getting the malaria. There are vaccine efforts underway, but the problem is getting to rural areas and getting them the boosters that they need. So that has been a huge barrier. And mosquito abatement is what we focus on the most. So we provide nets, we provide, education on mosquito control, reducing standing water environments and things like that, as well as, bug sprays and whatever to prevent mosquitoes from biting you, because they don't just transmit malaria, right? They can transmit a lot of diseases. Malaria, the way that it presents as a disease, we have a cycling of symptoms, which can be very crippling. We're talking about crippling pain and muscle aches. Cycling every 48 to 72 hours, depending on which version of malaria you have. And that coincides with the life cycle of those malaria protozoa. Basically, they live inside of your red blood cells, and then they burst out. And when they burst out is whenever we start seeing the inflammation and the signs and symptoms. And they'll go on with their life cycle. And every time they burst out, we'll have severe fever and inflammation associated with that. All right, talking about the hemorrhagic fevers. All hemorrhagic fevers are characterized by fragility of blood vessel walls, leading to bleeding. This could be literally bleeding out of the body, like through eyes and other orifices, or it could be petechial rashes, which are burst capillaries under the skin. And so these are all associated with the hemorrhaging in some way, and that's due to the fragility of the blood vessels. But they don't just have bleeding. Fever is also a thing. They also have fever. So this is the thing. If you have a cardio or lymphatic disease that has a fever associated with it and bleeding is associated with it, that's a hemorrhagic fever. If it's just the fever, but not typically the bleeding, then it's a non-hemorrhagic fever. Okay, so that's the difference there. Our hemorrhagic fevers include Ebola and Marburg. Both of these are viral infections caused by a type of virus that has a shape like, it looks like this. It's a shepherd's crook. Most of our viruses, remember, they're shaped like this or they're shaped like that. So this guy has a weird shape to him. These are called filoviruses because they look like filaments. And these, usually we think Ebola and Marburg are probably harbored within bat populations and are transmitted by blood to blood. Dengue and yellow fever and chikungunya. All of these guys are transmitted by mosquitoes. So dengue fever, yellow fever, and chikungunya are all hemorrhagic fevers that are transmitted by mosquito bites. We do have chikungunya endemic here in the United States. Chikungunya is a hemorrhagic fever that you can contract in the United States, primarily in Florida and the southern parts of the United States. We do have outbreaks of this, and it is tracked by the CDC here in the United States. We've also had increasing infiltration with dengue as weather warms and global warming is happening, and we have increased temperatures further and further north. We've had cases of dengue popping up in Texas and Florida and other regions like that in our mosquito populations. And we don't have yellow fever here in the United States, but it is present in other areas of the world. And then Lassa fever, which is maintained, I believe, in rodent populations and is not common. You don't usually get hemorrhagic fever from this, but it can lead to very severe hemorrhagic fever in some cases. All right, moving on to our non-hemorrhagic fevers. Most of these guys are I say TIC, it should be TICK, but tick-borne. So this is also another biting insect. So these are technically arboviruses or arbobacterium or arboprotozoa. All right, most of these guys that we're going to talk about here are bacteria, and I think there's one protozoa. So not many viruses in this list here that we're about to go over. It's mostly bacteria. All right, so Rocky Mountain Spotted Fever, RMSF. This is caused by the bacteria Rickettsia rickettsii, transmitted by tick bites. All rickettsial bacterial infections transmitted by ticks this way present pretty similarly to these spotted fevers. So Rocky Mountain Spotted Fever specifically is Rickettsia rickettsii. Here we have a pretty significant rash that'll present very characteristically on the body. And that rash, the lesions from the rash can actually merge together because it is a petechial rash. and it can lead to what we would consider to be a proper hemorrhagic fever, though that's less common. More often, you're going to start with the fever and the malaise associated with it. Rocky Mountain spotted fever actually has about a 20 to 30% mortality rate. It is no joke, and it must be treated with antibiotics. Next is Lyme disease. Lyme disease, also transmitted by ticks, is caused by bacteria Borrelia burgdorferi. Usually have a very characteristic bullseye rash associated with this, and very slow progressing disease, often neurological in nature. The Borrelia burgdorferi bacteria that causes Lyme disease, it can be harbored for a very long period of time without causing presentation of disease, and lives inside of the cells and can be very difficult to diagnose as well as treat. So this can be one that can cause chronic low-level disease presentation that people often don't know what the cause is and wouldn't link it back to a tick bite. Next is ehrlichiosis and babesiosis. Very similar presentation of disease between ehrlichiosis and anaplasmosis as well, which is another bacteria. Ehrlichia and anaplasma bacteria cause their associated diseases. Babesiosis is caused by a protozoa, but these are all very similar in how they present with the fever. Pretty mild. You often won't have very severe disease associated with this, just mild fever, maybe some malaise and fatigue. And you can often recover from these diseases pretty naturally. However, babesiosis can last a little bit longer because it is protozoal and might need treatment with antiprotozoal drugs. Other than that. All right, moving on to Chagas disease. Chagas disease is caused by the protozoa Trypanosoma cruzi. And so one of the trypanosomes that we looked at in our microscopes was Trypanosoma gambiense. So Trypanosoma gambiense is another cardio and lymph disease that's associated with African sleeping sickness. And that can lead to what it sounds like. It causes just almost like narcolepsy, sleeping, even whenever operating heavy machinery, and that can cause issues, but it can also lead to severe inflammation of the heart. Trypanosoma cruzi, however, is also a protozoa. It's a flagellated protozoa. It's transmitted by the bite of a triatomine bug. These are also known as kissing bugs. These are endemic here in Oklahoma, and these bugs can harbor Chagas disease even here in Oklahoma. Much more common in Texas and in the true South. But these kissing bugs, they're everywhere. They live in your house, I bet. Like, I promise you, they live in your house, and you probably haven't seen them. They're about an inch to an inch and a half in size, and they bite, and they lap up the blood. They don't suck it through a proboscis. They lap up the blood, and then they defecate, and then the protozoas can actually migrate in through the skin. The site where they migrate in can cause inflammation called a chagoma. And so that's where Chagas disease comes from. That's where that word comes from. And then this can lead to neurological as well as systemic disease, fever, inflammation, and neurological effects from that. More common in little babies because they are less prone to be able to swat bugs off of them that are biting them. So we see this more commonly in infants and children who live in like cribs and stuff like that, that sort of age range. HIV. HIV stands for human immunodeficiency virus. You cannot have AIDS without HIV, but you can have HIV without AIDS. AIDS is basically defined by how many T cells you have. Once you have lost a certain number of T cells, then we consider you to have AIDS. And AIDS is associated with opportunistic infections, though even low-level HIV infection can lead to opportunistic infections. You also will have fever and malaise just associated with having the HIV infection. HIV infects CD4 positive T cells specifically. If you have CD4 on you, then you can be infected. So this is your T helper cells, your Th1s and your Th2s. So once your Th1 and your Th2 counts start going low, you can't activate your antibody and you can't activate your CD8 cytotoxic T cell killer cells. And so that basically leads to very severe, significant immunodeficiency as a result of that. HIV as a virus has 3 proteins that it brings with it. Reverse transcriptase, which turns its single-stranded RNA genome into DNA. Then integrase, which puts that new DNA into the host DNA. And then protease. When the virus makes its proteins, it makes it in one long protein, and then protease cuts it into the parts that it needs for the capsid, for the reverse transcriptase, for the integrase. The protease cuts all that down. And so we can block any of these proteins, the reverse transcriptase, integrase, or protease, with our antiretroviral drugs as targets. Finally, we're on chapter 22, These are our respiratory diseases. All right, so in our respiratory system, our natural first line defenses will be our nasal hairs, the cilia in your lungs, the mucus to trap things, and also in our respiratory tract, in your lungs, you have alveolar macrophages inside the alveoli, the little sacs that fill with air where we have oxygen and gas exchanges happening. Here we have also production of secretory IgA in our mucosa of our respiratory tract. This will be in your mouth, in your sinuses, and in the actual lung tissues and stuff like that. We will have this antibody there to help with defending against microbes. All right, normal biota of the respiratory tract can include things that can cause disease. This includes strep pyogenes or Haemophilus influenzae, which can cause the meningitis, or Haemophilus influenzae is also associated with pneumonias and things like that. Streptococcus pneumoniae, which in its name tells you it causes pneumonia, but was also our #1 cause of meningitis. Even Neisseria meningitidis, our worst case meningitis bacteria, even that can be harbored in your respiratory tract naturally. Often in your sinuses is where these things will live naturally in small, small numbers. When they progress down into your lungs or migrate into your nervous system is when we start presentation with those diseases. And it can also include Staph aureus. That's our normal biota. So moving on to our diseases, common cold, which we call rhinitis. Acute rhinitis is the common cold. If you have allergic rhinitis, I'm going to take my jacket off, I'm a little warm. Allergic rhinitis is if you have inflammation of the nose, which is what rhinitis means, caused by allergies. But when we talk about infectious cause of this, oftentimes viral rhinitis is the common cold. There's over 200 different viruses that can cause the common cold. 200 different viruses. So when we say you have the common cold, we're really talking about a constellation of signs and symptoms, right? You may have malaise, fatigue, you'll feel like junk, you might be, tired, you might be sore, you might have a runny nose, your eyes might be red or watery, and things like this associated with having a cold, maybe a low-grade fever. So those are signs and symptoms associated with a cold. So this is a syndrome. And then if you have a common cold, it can be caused by any of these 200 viruses. And this even includes RSV, which we're going to get into in a little bit. But adults are more common to present with common cold symptoms from RSV, whereas younger children, especially 6 months and younger, are going to have much more severe respiratory disease associated with it. Common cold is also commonly associated with secondary infections, which can include sinusitis, which can be caused by viral infections more commonly than you think. Sometimes can be associated with secondary bacterial inflammations. You have inflammation caused by the viral infection. Bacteria take hold and take advantage of that and can overgrow. And this is actually what usually leads to causing otitis media, middle ear infections, usually from inflammation of the eustachian tubes, improper drainage of the fluid associated with the middle ear, and then the bacteria or the yeast can overgrow, bacterial or yeast in nature. All right. Pharyngitis, inflammation of the pharynx, which is your throat. This is often related to a lot of different bacteria and viruses can cause pharyngitis, but one of the ones that we're most familiar with is strep throat, caused by Streptococcus pyogenes. Again, the toxins that it produces often are caused due to lysogenic conversion. A phage gave them these toxins. Many of its other virulence factors help in causing the disease, which we talked about in our skin diseases. Complications can include that skin presentation of scarlet fever or even rheumatic fever, where we have inflammation in the cardiovascular system. And then it can even lead to that acute post-streptococcal glomerulonephritis, which we learned about in chapter 17, is a type 3 hypersensitivity. Basically, the antibodies to the strep bacteria combine with strep antigen and get trapped in your glomerulus in your kidney and lead to inflammation of the kidney that can be damaging and even lead to permanent kidney damage. Some pharyngitis also can be caused, bacterial pharyngitis can sometimes be caused by Fusobacterium necrophorum. Fusobacterium necrophorum, which can be part of your oral biome. Fusobacterium necrophorum can be associated with peritonsillar abscesses, which can be life-threatening and can even lead to septicemia if it goes unchecked. All right, moving on to whooping cough. Whooping cough, also known as pertussis, is caused by the bacteria Bordetella pertussis. There is vaccine available, the Tdap or DTaP. It just depends on what version you're getting. But Tdap, whatever it is, it often comes with tetanus, diphtheria, and pertussis. I'm not really going to talk too much about diphtheria. Diphtheria was caused by, it's another upper respiratory throat disease caused by Corynebacterium diphtheriae. And Corynebacterium diphtheriae also makes a toxin called diphtheria toxin, which can lead to presentation with pseudomembrane in the throat. And so literally it creates a membrane with its bacterial growth and its biofilms and everything that closes off the throat completely. And so that would be diphtheria. With pertussis, though, coming back to that, Bordetella pertussis, the disease presentation occurs in 3 phases. The first phase is the catarrhal phase. This is where you'll have cold-like symptoms and what we might consider to be the prodrome. Remember the prodrome when we talked about our chapter 13, evolution of our diseases? The prodrome is when you have your first signs or symptoms. Next phase of pertussis is the paroxysmal phase. This is where you have the uncontrollable cough. Pertussis, also known as whooping cough, was also known as the 100-day cough. And if you've ever had one of those things where you accidentally breathed in a little bit of water and you just can't stop coughing and then can't quite catch your breath and you're like, like that, that breath in is the whoop of whooping cough. Imagine that for 2 or 3 months straight. And it's just that. And there is a video associated with this lecture that I do encourage you guys to watch where a little baby has this. We actually see presentation of pertussis more commonly in little newborns where they're going to be more susceptible to it. They're not vaccinated yet and can be very severe in newborn babies. Then after that uncontrollable cough phase, that uncontrollable cough actually causes severe damage to the cilia, which help work up the, basically, if you breathe something in, any bacteria, any particulates or whatever, it gets trapped in your mucus, and your cilia in your ciliary tree of your respiratory tract help move everything out, and then you cough it out. But when you have pertussis, that becomes so damaged that that doesn't work correctly anymore. And so that can actually lead to very severe secondary infections as a result of this. So you might have cleared the pertussis bacteria, but you're not out of the danger yet because of secondary infections. Again, vaccine is very effective against preventing this. RSV, also known as respiratory syncytial virus. Respiratory syncytial virus. Very characteristic presentation of cells merging together, which we call syncytia, multinucleated cells as a result of this. Usually in otherwise healthy adults will have cold-like symptoms. However, in children that are 6 months or less, this presents as very severe respiratory disease, cyanosis, and things like this that will require usually antiviral treatment. Moving on to influenza. Influenza is a viral disease caused by the influenza virus. Influenza viruses are characterized by their H and their N. So like, for example, H1N1 or H5N1, it just matters what kind of H or what kind of N you have, and that's how we characterize flu. The H stands for hemagglutinin. It is our attachment receptor that helps us attach to our target cells and also helps blood cells stick together. Neuraminidase is an enzyme that helps with budding, fusion, and breaking down mucus. Tamiflu, oseltamivir, as well as, I believe, Relenza. These guys affect the neuraminidase activity of flu and basically keep the neuraminidase from helping with budding, helping with breaking down the mucus and things like that. However, you want to give it early on in the infection because once the virus establishes itself, the antiviral just isn't effective at controlling that very well. Flu can mix and match its H's and its N's, and it has 8 different segments in it. So if this is your virus here, there's a whole bunch of gene segments in it. There's 8 of them. And it can basically, like if I get infected with one virus and also happen to get infected with a different flu virus, while my cell is infected with these, I can actually make a whole new virus as a result. If I get infected with 2 viruses, the virus that comes out of it can have part of the original and part of the other virus. And so when this happens, when we have this sort of gene reassortment, we call this antigenic shift. And I'm use language again. I tell you guys I'm trying to work on my bad language, but this is a time when I feel like it kind of applies. So I'm going to use the words. Okay. So I was teaching this one time and I accidentally said shit instead of shift. And you could look at it this way. If you have this happening in your body where you're mixing and matching viruses going on, that is an oh shit moment. But we also see this happen a lot in pig populations and swine. So swine can become infected with a bird virus and a human virus. And now suddenly you have a bird plus human hybrid. And pigs are very poopy animals. I think we can agree. So they're poopy, and this is a oh poop moment. So to me, that's how I remember that's the antigenic shift. Flu can also go through something called antigenic drift. This is just normal genetic mutations. Every flu season, when we come out with new flu vaccines, those new flu vaccines usually are dependent on antigenic drift. So we'll see some minor mutations. Each time that flu has a new mutation, your body sees it as a whole new virus. So we have to have new vaccines for those whole new viruses. They might as well be completely different viruses to your body, to your immune system. Once we have that's enough. But when we have antigenic shift, the oh shit moment, right? When we have that happening, that's such a new virus. There's no part of this virus often that looks even new to you. So while you might get the normal flu from antigenic drift viruses, when you have an antigenic shift like this, that looks like a brand new, you have no defenses whatsoever to any part of this virus. So this is where we see pandemics. This is where we see very significant increase in disease in human populations. It's from antigenic shift. Like I said, oh shit moments. So that's the flu. Moving on to tuberculosis. Tuberculosis is a bacterial disease. It's caused by the bacterium Mycobacterium tuberculosis, which we learned is acid fast because it has mycolic acid wax on its surface, which our carbofusion, would bind to that wax because it was lipid soluble and stain our mycobacterium, that fuchsia color, in the lab. So Mycobacterium tuberculosis is the major cause of tuberculosis. Tuberculosis can also be caused by other mycobacterium species like Mycobacterium avium in people who are immune compromised, but they're still mycobacterium species. This is diagnosed usually by the tuberculin skin test, which is that PPD skin test where they inject a little bit of the antigen under the skin. And then a couple days later, they see if you've had a reaction to it. If you have had a reaction to the PPD skin test, it takes a couple of days because that is a type 4 hypersensitivity. Your CD8 T cells are what respond to that. And it takes a little while for them to resupply their numbers. And so it's a delayed response. So it's delayed type immunity. This is because Mycobacterium tuberculosis lives inside of your alveolar macrophages. It's not an outside bacteria like staph is. Mycobacterium tuberculosis actually lives inside of your alveolar macrophages. So while your macrophages are out there eating up stuff, phagocytosing everything, the Mycobacterium tuberculosis bacteria says, hey, thanks for the house. This is exactly where I wanted to live. It basically protects itself from that phagolysosome fusion during that phagocytosis aspect, but lives inside of the alveolar macrophages. So the tuberculin skin test is testing for that inside response, your CD8 response, seeing if you have one. We can also diagnose it with chest x-ray, where we will see clouding in the chest in the areas where the bacteria has caused damage, because the bacteria living in those alveolar macrophages cause tubercles. This is basically like where we'll have your white blood cells walling off the infected alveolar macrophages and the bacteria in a granuloma. It's a granuloma. And then that granuloma becomes caseous lesions, which is cheese-like, and then can become even necrotic or even calcify and harden over time. And so we can have developing of these tubercles as a result of your primary infection. But you might not even present with very severe respiratory symptoms during this phase. You might even be asymptomatic during this phase. It's whenever this reactivates with secondary tuberculosis that we see true tuberculosis, where you're coughing up green and bloody sputum, where you have consumption, where you are anorexic, and all of the presentation that we typically associate with tuberculosis and consumption. That happens with reactivation tuberculosis. So you can actually be asymptomatic for quite a long time. But those tubercles can be visualized with the chest x-ray. And of course, we know acid-fast stain of the sputum. doing that acid fast stain to look for the mycolic acid. If we see mycolic acid, you have Mycobacterium tuberculosis or avium, leading to tuberculosis. And so it's a pretty easy way to diagnose this without having to culture these guys. We did talk about the skin test. Yes. Okay. There is a vaccine available for tuberculosis. It's not as widespread as I feel like it probably should be, considering that a third of the world actually has tuberculosis even right now. Quite a shocking number, really, when you think about it. Oh, yeah. So one thing I want to say before I move on, HIV, really quick, because that reminded me. HIV. I think I might have said it in class before, but I just want to drive this home, y'all. Half of all sexually active aged adults. So if you are, 16 or older, in your sexual prime from 16 to like about 45 years old. Half of all of those adults in South Africa have HIV. Half. We think it's a problem here, man. All right, so back to our respiratory diseases. Now we're at pneumonia. Pneumonia is a syndrome. It's characterized basically like its constellation of signs and symptoms associated with it. Fluid in the lungs, maybe cyanotic. You might have fever. You'll be tired. You'll have a hard time getting enough air. You'll have a hard time breathing, shortness of breath, chest pain, and weight on the chest, and all of this stuff associated with pneumonia. Most commonly true pneumonia is caused by Streptococcus pneumoniae bacteria. Also common cause would be viruses and other things like that. Bacterial pneumonia most commonly caused by Streptococcus pneumoniae. If you have community-acquired pneumonia transferred from person to person, just generally in normal situations, it's usually Streptococcus pneumoniae. Again, viruses are also pretty common. If you have walking pneumonia or atypical pneumonia, this means you still are getting that fluid, you're still getting that bacterial growth in the lungs and fluid buildup in there, but you're not hospitalized from it. You're still able to function. And so that walking pneumonia is usually caused by Mycoplasma pneumoniae. It's a different presentation of pneumonia. And remember, mycoplasmas, the plasma, it's not mycolic acid this time. The plasma aspect is what's important here. These are the ones that do not have a cell wall. Remember those guys, the tenericutes. Back to chapter 4. Other microbes can also cause pneumonia, not just Streptococcus pneumoniae, but other ones that are less common would be Legionella, leading to Legionnaire's disease, hantavirus, which is a viral version. Hantavirus causes hantaviral pulmonary syndrome, and it's basically transmitted by the urine and feces that have dried up from infected rodents in general, but mice are more common, deer mice, and can lead to pretty severe pneumonia and pulmonary symptoms, and even hemorrhagic fever. Haemophilus influenzae can lead to pneumonia as well. And then Histoplasma capsulatum, which is going to be a fungal infection. This is the one like they are associated with like bat droppings or caves and things like that. But people who are immune compromised can get this just from an environment. So it is a fungal pneumonia. All right, healthcare associated pneumonias are also pretty common. Healthcare associated pneumonias are usually caused by different kinds of bacteria, including Klebsiella pneumoniae and other things that are usually actually harbored in the gut or in the environment around us. But there's a lot of different bacteria that are associated with healthcare. About 1% of all patients who are hospitalized will get healthcare associated pneumonia. If you're hospitalized and put on a respirator, 1% of the people who are receiving respiratory care in a hospital will get pneumonia. But 50 to 70% of those people will die from that pneumonia if they get it, hospital-associated pneumonia. So it's really important maintaining as much cleanliness as possible for respiratory care, changing the tubes, aspirating any fluid in the lungs, encouraging the patients to sit up if they can, encouraging them to cough if they can, and keeping as much of that airway clear and clean and as sterile as possible in preventing that. All right, I read chapter 19. Why are we back at it? I think I put it in here twice. Okay. Let's go over our most commonly missed topics. First one is going to be gas gangrene. So remember, gas gangrene is caused by Clostridium perfringens, which is gram-positive, endospore forming, and it is anaerobic. It causes what we call anaerobic cellulitis or true myonecrosis if it spreads within the nearby healthy tissue. It has lots of active toxins that are associated with its disease. So gas gangrene, again, caused by Clostridium perfringens, which is an endospore-forming gram-positive anaerobic bacteria. A common types of questions that I will ask. There's a lot of diseases we got to know, right? And a lot of causes of them. I have a chart that is posted in you guys's modules that has like all of the diseases, the names of the organisms that cause the diseases, and then whether they're virus, fungus, protozoa, or bacteria. Common questions will be, what type of microbe causes gas gangrene? You should know it's a bacteria. I could also ask, which species causes gas gangrene? And it might say Clostridium perfringens, Clostridium botulinum, Staphylococcus aureus, and you have to know which of those species causes the disease. So those are all common. They're very basic, but there's a lot to know in these chapters. So there's a lot of questions like that for Unit 4. Next, why does it require deep wounds to cause the cellulitis or myonecrosis that is associated with gas gangrene? Because Clostridium perfringens is anaerobic. Doesn't do well in exposure to oxygen. So is this because all endospores always require deep wounds to be reactivated? No. Bacillus species don't need to be in deep wounds. This isn't because of the endospore aspect. Is it because nutrients, the nutrients the microbe needs can only be found in deep tissue? It's not because of that. The causative organism only makes its toxins in anaerobic, oxygen-low environments. Yes, anaerobic environment is why they need to be in deep tissues. The organism is sensitive to light. It's not the light, it's the air. Next, impetigo. Impetigo is caused either by S. aureus or S. pyogenes. It's usually associated with S. aureus's exfoliative toxins, as well as the fact that S. aureus also makes coagulates, which coagulates plasma and blood. Strep pyogenes can also be associated with it. It is beta hemolytic and can lead to all these other things as well. All right, so which organism is associated as one of the causes of the disease impetigo? For impetigo, it was S. aureus or Streptococcus, Staph aureus, I'll even put that, Staph aureus or Streptococcus pyogenes. Let's see, is this in our list? Streptococcus pyogenes, yes. Was Staphylococcus epidermidis? No. Streptococcus saprophyticus? No. Staphylococcus empiricus? No. So this is what I'm saying. You got to know your organisms. Common types of questions on the exam. A culture grown from a 5-year-old skin sample shows that it has visible irregular clusters of cocci. Irregular clusters is staphylo. Nat is flying at me. And cocci is round. Is this consistent with a typical case of impetigo? So if our two causes can be Staphylococcus aureus or Streptococcus pyogenes, which is this, does that align with either of those? Yeah, Staphylo and Coccus, right? So it's going to align with Staphylococcus aureus. So does it match the description of both typical causes of impetigo? No, it doesn't match the strepto, which is the other cause. It matches the description of one of the typical causes, yes, which is Staph aureus. The other ones just kind of negate themselves out once you get to there. Okay. Chapter 20 are nervous system diseases. Fungal meningitis. Our two major causes are going to be Cryptococcus neoformans, which we often see in the immunocompromised, or Coccidioides immitis, which is usually associated with valley fever. So we'll write that. Fungal meningitis, usually associated with Cryptococcus neoformans, which is usually going to be immunocompromised, or Coccidioides immitis, which is usually associated with valley fever. which is normally in like Arizona. Okay. All right. So which of the following is a causative agent of fungal meningitis? Is it Clostridium perfringens? No, that's gas gangrene as well as food poisoning. Is it Cyclospora? No, that's diarrhea. We haven't gotten to that, but that's chapter 23. Is it Coccidioides immitis? Yes, that was the valley fever one. Is it Crimean Congo, HFV, which is hemorrhagic fever virus? Well, I would hope that a virus wouldn't be a cause of meningitis, but Crimean Congo hemorrhagic fever virus is one of the hemorrhagic fevers. And it also happened to be what I studied when I was in graduate school. Which form of fungal meningitis is usually associated with AIDS? This is an immunocompromised situation. That was the cryptococcal. Cyclospora, again, diarrhea. Coccidioides immitis associated with valley fever. And Clostridium perfringens, again, gas gangrene. All right, amoebic meningoencephalitis. Inflammation of the meninges, and we're having inflammation of the brain associated with an amoeba infection. The two different ones were Naegleria fowleri, which was associated with PAM, primary amoebic meningoencephalitis. And then the other one was GAM, which was acanthamoeba. All right, so PAM, Naegleria fowleri, and GAM, acanthamoeba. Both of these are protozoa, and both of these are amoebas. They move by pseudopods. All right, so which of the following pathogens is an amoeba? Well, we know Neisseria gonorrhoeae is bacteria. We'll learn this in chapter 24, but Neisseria should give it away. We've already learned about Neisseria meningitidis, which is a bacteria. Neisseria gonorrhoeae is another bacteria, sexually transmitted disease. Alcaligenes faecalis. This is also a bacteria. Naegleria fowleri. This is our amoeba that causes PAM. See? Erythema infectiosum is actually not an organism. It's also known as Fifth's disease. It's a viral infection. Which of the following pathogens is the cause of PAM? So PAM is an amoebic meningoencephalitis. So we already know it has to be an amoeba. The only two amoebas on here are Naegleria fowleri and acanthamoeba. Giardia is not an amoeba. It's a protozoa, but it's not an amoeba. It has flagella. And it causes diarrhea, not meningoencephalitis. Trypanosoma cruzi. Trypanosoma cruzi is a flagellated one as well. Trypanosoma cruzi was associated with Chagas disease, not meningoencephalitis. So it has to be Naegleria fowleri or acanthamoeba, and this is just a matter of knowing which one is which. Naegleria fowleri was PAM, and acanthamoeba was GAM. All right, our arboviruses. Arboviruses are viruses that are transmitted by biting insects. We have some diseases listed here in our little review. West Nile virus, St. Louis encephalitis, toga viruses like Eastern equine, Western equine, Venezuelan equine, which you might be like, oh, equine viruses, who cares about these? No, these also infect people and can be very deadly. And then the Bunya viruses like California encephalitis or lacrosse viruses and some other viruses that fall into that category. All right, so what is a primary transmission method of arboviral encephalitis? These are biting insects. It's not airborne, it's not fomites, and it's not transplants. It's going to be mosquito bites. What is encephalitis? Itis is inflammation. Encephala is the brain tissue. So is it inflammation of the skull? Not the skull, the brain. Is it infection in the nerves? It's inflammation in the brain. Is it inflammation of the brain? Yes. Is it infection in the brain? No. Believe it or not, you can have infection without inflammation, and you can have inflammation without infection. So inflammation in the brain is encephalitis. We just happen to be talking about infectious causes of that inflammation. So don't get tripped up on stuff like that either. Chapter 21, the plague. The plague, again, caused by the bacteria, Yersinia pestis. Reservoir in rodents transmitted by flea bites. The bubonic plague is the one that's found in the lymph nodes. The pneumonic plague, the respiratory system, and the septicemic plague, where it is flourishing in the bloodstream, is the deadliest one. We're talking 90% mortality rate. Bubonic plague is the one you want to get if you're going to get the plague, by the way. It's only like a 20% mortality rate. Bubonic plague, a bubo, is a swollen infected lymph node that's actually been infected in bubonic plague. So there's a little chart here to help with breaking that down. Which of the following choices represents the type of plague referred to as the Black Death? When you actually have necrosis and death of tissue, especially in the extremities, this leads to it turning black. This is what we see in when it becomes systemic, and it's systemic because it's being spread and flourishing in the bloodstream. So that is septicemia, not cutaneous. We don't have a skin-borne version of it. Bubonic is just in the lymph nodes, and pneumonic is just in the lungs. So when we have actual Black Death, we're talking about that systemic necrotic black tissue. Which of the following choices represents the type of plague involving infected and inflamed pus-filled lymph nodes? If the lymph nodes are the target there, those infected, inflamed, pus-filled lymph nodes are actually called buboes. And so this is the bubonic plague. Pneumonic is in the lungs. Cutaneous isn't one of them, but that would be in the skin is what that means. And septicemic is in the bloodstream. Next is malaria. Malaria is a protozoal blood disease caused by an apicomplexan plasmodium, transmitted by mosquito bites. We want to target mosquito prevention methods and using drugs based on quinine to treat the disease. So this is a protozoa, Plasmodium falciparum is the big one. There's other plasmodiums like Plasmodium vivax. There's other ones too. Plasmodium species. That's how we abbreviate that. All right, so which type of pathogen causes malaria? Is it a bacteria? No. Is it an archaea? No. Is it a helminth? No. It's a protozoa, and a protozoa is a eukaryote. So it's not a helminth, and I didn't put protozoa as a choice, but we should know protozoa are eukaryotes. How is malaria transmitted? Mosquito bites for sure. It's not person to person through sex. It's not intact skin. It's not fecal oral. It is mosquito bites. All right, chapter 21. Our non-hemorrhagic fevers. Focusing on the difference between Lyme disease and Rocky Mountain spotted fever. I am going to point something out to you guys. Rocky Mountain spotted fever starts with an R. Rocky Mountain spotted fever starts with an R. This is Rickettsia rickettsii. Lyme disease doesn't have a B in it, but it's Borrelia burgdorferi. Notice there's no B in RMSF, okay? So that's how I kind of just remember those. Borrelia burgdorferi. Babesiosis is caused by Babesia. Ehrlichia is caused by Ehrlichia. Anaplasmosis is caused by Anaplasma. They're in their names. Lyme disease and Rocky Mountain spotted fever, it's not in the names. But RMSF, R. Rickettsia rickettsii. Try to remember that. Okay, so I'm going to write that down here. RMSF, Rocky Mountain spotted fever, R. rickettsii. Lyme disease, also, they're both transmitted by ticks. And they both cause non-hemorrhagic fevers, typically. Lyme disease is B. burgdorferi. Okay. Which disease does Borrelia burgdorferi cause? It's not RMSF, because that is the rickettsia one with the R. It's not babesiosis, because that's caused by Babesia. It's not ehrlichiosis, because that's caused by Ehrlichia. It's Lyme disease. Which organism causes Rocky Mountain spotted fever? Rickettsia. Anaplasma phagocytophilium causes anaplasmosis. Ehrlichia chafensis causes ehrlichiosis. Borrelia burgdorferi causes Lyme disease. All right, finally, chapter 22, going over whooping cough and the different phases. We have the incubation phase, which is no symptoms yet. It's just the time that it takes from exposure to the bacteria to the prodrome, which is our initial symptoms. The catarrhal phase, this is our prodrome. First signs and symptoms. Paroxysmal, cough. And convalescent is recovery. But we can also have, during that convalescent phase, increased risk for secondary infections. Because we've damaged our cilia. Okay, so what type of pathogen causes whooping cough? Whooping cough is caused by the bacteria Bordetella pertussis. So it's bacteria. Anybody that knows anything about kennel cough will tell you that kennel cough is also caused by Bordetella bacteria, but in like dogs and stuff like that. So it's all different kinds of Bordetella that can cause kennel cough, but it's still Bordetella. Bordetella gives you coughs, man. So for humans, Bordetella pertussis is the bacteria that causes that. It's not a virus, it's not a protozoa, it's not a fungus, it is a bacterium. And that toxin, that pertussis toxin, is what's leading to all these bad effects. How is the paroxysmal phase of pertussis defined? So when we're talking about catarrhal, paroxysmal, and then the convalescence. So I feel like I had a thing. Oh, I can't play chess carefully. All right, so incubation, catarrhal, paroxysmal, and then convalescence. And just also reminds you of being careful for that last convalescence because you're going to be more likely susceptible to infections. So this is our order here. So the paroxysmal phase is the cough. This is just cold-like, cold, cough, recover, but susceptible to other secondary infections. So cough, it's not the cold-like, that's the catarrhal. It's not, it is the severe coughing fits. Decrease, this is the convalescence. And obviously death, I would hope that doesn't happen. I think this is the last one. Oh, we got two more. Okay. Mycobacterium. This is tuberculosis caused by the bacterium Mycobacterium tuberculosis that has that mycolic acid, which we can see in our acid fast state. We kind of just went over this, so I'm not going to go into too much more detail here. But the bacteria Mycobacterium tuberculosis lives inside of the phagosome of the alveolar macrophages, inside the alveoli of the lungs and these little sacs in the lungs. And y'all, this was one of my attempts to try to make an AI image here. What is this? What's these? This is why AI can't be trusted, y'all. It doesn't know words. This. And I try to tell it to spell the words correctly, and it can't. It doesn't know what's going on in these images. So don't trust AI to make your images, y'all. It does this crap to it. But the image is still effective. In these sacs inside your lung, the bacteria will get down into those sacs, and then the macrophages that are down in those sacs eat them up, and then they are happy to live in there. Tuberculosis is caused by Mycobacterium tuberculosis, which is a bacteria. It has mycolic acid wax and is acid fast and lives in the alveolar macrophages. So what molecule present on the surface of Mycobacterium species is unique to the species and is used as a common identifier for diagnosing purposes? On the surface of Mycobacterium, this is mycolic acid, which is that wax on the surface, which we identify with our acid-fast stains. Not LPS, which is only on gram negatives, not endotoxin, which these are the same thing, by the way. Gram negatives. And it doesn't make endospores. That's only going to be Bacillus or Clostridium. All right, what type of pathogen causes tuberculosis? I mean, at least this one kind of is in the name, Mycobacterium. So this one shouldn't be too hard. This is a bacteria. It's not virus, protozoa, or fungus, but people still manage to miss this one. So be sure you know that. All right, influenza, the flu, caused by a virus we have the hemagglutinin for binding. We have the neuraminidase for breaking through the mucus and helping with budding as well as entry into the cell. Tamiflu affects that neuraminidase. And then we have antigenic drift, which is random natural mutations just from making so many viruses and accidentally making mistakes with that. Or antigenic shift, which is the reassortment of those gene segments, creating that whole new mixture of virus. So it says down here, drift is small, shift is huge. To remind us that. Shift equals huge. Gene reassortment, segment reassortment inside a single host. When a pig gets infected with 2 different flu viruses and mixes and matches them, and a new one comes out. Drift will be random, like you're drifting in the breeze. You're just slowly making your way along, making random mutations, but a big shift, huge change. All right. So what is the term for the alteration of the influenza virus genome due to minor errors in the genetic sequence? This is the antigenic drift, not antigen drift, which I won't make it that close on the exam, but it's antigenic drift. Oh, whoops. What is antigenic drift? Is that whenever mutations in the genome, I say basically it's already over here. Antigenic drift, is it from mutations in the genome that the virus can have happen randomly? Yeah. Segments of the flu genome reassort within the host. This is shift. Flu virus obtains an extra gene segment. That's not a thing that happens. When the flu virus loses one of its, it doesn't happen. It has to have all 8. It has to have 8, but those 8 can come from different viruses. If that happens, those 8 coming from different viruses, that's shift. But when we have random mutations, that's drift. Okay, that is really it. Have any questions? All right, if you guys get any questions when you're studying, email me. Otherwise, I'll post this video today or tomorrow, and I will have a video for chapters 23 and 24 up by the end of Monday. It'll probably be sooner than that. So that when we're ready for our review on Wednesday, you guys will be all prepared and ready for that. All right, if that's it, everybody have a good day, have a good weekend, and I'll see you guys on Monday, either via Zoom or I will be here if you guys want to come here. Okay.
You like expected to memorize the whole list of unit 4 diseases, just like the ones that are covered in the objectives.
The ones that are covered in your objectives.
Like the MCRO and the...
So there's a lot, right? There's a lot that are in like the textbook and some that are on that chart that I have too. But the only ones I will ask questions about are the ones in your learning objectives. So, but if I say like, know the organisms that can cause pneumonia, then you need to know all of them that can cause pneumonia. You know what I mean? But if I'm talking about specific ones, then I expect you to know the specific ones. But if I don't mention something like, I don't think diphtheria is in your learning objectives. So I don't think you need to know. I literally write my questions from the learning objectives. So if it wasn't in the learning objective, I don't have a question for it. So that's pretty much where it's going to come from because I literally write them from that. Yeah. So yeah, so it's not in the learning objective. Don't bother learning it. Cut it out. Yeah. Good question.
I think for a unit 3 exam, I really just went based off on the learning objectives and did better on it.
Yeah, it helps you cut it down a little bit so you know what to focus on as opposed to, yeah, so focus on the learning objectives because I literally write the questions based on that. I sit there with the learning objectives and I'm like, okay, let's write a question. We need to explain this. So how can yeah, that's how I write them. So I'm not going to pull from anywhere else. Yeah.
I'll see you.
See ya.